Now we start our botany practical and Viva session. The first session, that is the study of mitosis. The study of mitosis in onion root tip. Study of mitosis in onion root tip. What about mitosis? Mitosis occurs in somatic cells. After mitosis, one cell gives rise to two daughter cells, identical daughter cells with a diploid condition. There are mainly four stages in mitosis. There are mainly four stages in mitosis: prophase, anaphase, metaphase, anaphase and telophase. In practical section, we usually get metaphase slide because it is very clear. So, we can discuss the different stages of mitosis. First, in any case of cell division, mitosis or meiosis, there are mainly two sections. First, nuclear division known as karyokinesis, and second, cytokinesis or cytoplasmic division. We discuss about karyokinesis. The first stage, prophase. The prophase is divided into three, that is early prophase, middle prophase and late prophase. Altogether, at first early prophase stage, the centriole, that is the cell organelle, helps for cell division. The centriole divides and each of them start moving to two opposite poles. The centrioles divided into two and start moving towards the opposite poles of the nucleus of dividing cell. The chromosome appear as long threads. The chromosomes appear as long threads and start coiling. Nucleolus enlarges. Nucleolus enlarges and become less distinct. In middle prophase, the chromosome condensation is complete and they become short and thick. Each chromosome is made up of two chromatids held together at their centromere part. Two chromatids, that is the arm portion. Here we can see the chromatids. The two chromatids held together at their centromere portion, that is the middle centromere portion. Each chromatid contains newly replicated daughter DNA molecules. Each chromatid arms contains newly replicated daughter DNA molecules. Now complete middle prophase. Then late prophase. Centrioles reach the opposite pole of dividing cell. The centriole reached opposite pole of the cell. Some spindle fibers. Here we can see the spindle fibers. Some spindle fibers extend from pole to the equator. Pole to the equator. Some spindle fibers extend from pole to equator of the dividing cell. Nuclear membrane completely disappear. Nucleolus is not visible. Now complete the three stages of prophase: early, middle and late prophase. In early prophase, centriole divide to two and they move toward opposite poles. Chromosome condensation starts. Nucleus enlarge and become less distinct. In middle prophase, chromosome condensation complete they become short and thick. Each chromosome is made up of two chromatids held together at their centromere portion. Each chromatid contains newly replicated DNA molecule. Then late prophase centriole reach the opposite pole of dividing cell. The centriole reach at opposite pole of dividing cell. Some spindle fibers are formed. Nuclear membrane disappear. Nucleus is not visible. Then the next phase. But in this picture we just draw the typical cell. But there are a lot of changes occur in prophase stage. We already discussed the changes that take place in prophase. Then the second stage, metaphase. The chromosomes are brought towards the equator of the cell. The chromosomes arranged at the equator of the cell with the help of spindle fibers. The chromosomes are brought towards the equator region by the help of spindle fibers. Each chromosome becomes attached to the two spindle fibers. Each chromosome becomes attached to the two spindle fibers by centromere. Each chromosome becomes attached to the spindle fiber by centromere portion. Whereas each centromere is joined to the opposite poles. Whereas each centromere is joined to the opposite poles. The sister chromatids are not yet separated. In metaphase, the sister chromatids not yet separated because the centromere has not divided. In anaphase, that is the third phase, centromeres of all the chromosomes divide. Centromeres of all chromosomes divide, and then each chromatid become a chromosome. Centromere of all the chromosomes divide, and then each chromatid become a chromosome. Spindle fibers contract and pull the centromere to the opposite pole. The spindle fibers contract and pull the centromeres of the chromosomes to the opposite poles. As the chromosomes are pulled by spindle fibers to opposite poles by their centromeres, they look like V, J or I shape depending on the position of centromere. Here we can see V shape. Then cytokinesis begin at the late phase of anaphase. The cytoplasmic division start. A furrow. In case of plant cell plate is starting to develop at the center portion. That is phragmoplast. Phragmoplast or a cell plate start to develop at the center portion of plant cell. What about telophase? In telophase, chromosomes uncoil to form chromatin network like this, chromatin network as a parent nucleus. New nuclear membrane is formed. Around each daughter nucleus. Nucleolus again reappear in each newly formed daughter nucleus. Now complete the karyokinesis, that is the nuclear division. Then cytokinesis, that is cytokinesis means cytoplasmic division start at the latest phase of anaphase and complete at the case of telophase. There are two types of cytokinesis that is different from animal cell and also in plant cell. We discuss about the onion root, so it is plant cell. Here the cytokinesis or cytoplasmic division. Here it is a process of division of cytoplasm of a dividing cell into two. A phragmoplast or cell wall begin to develop at the central portion and develop to peripheral region. Then at the last stage of telophase, the cell become completely divided into two by the development of cell part or phragmoplast. Phragmoplast or cell plate divide the cytoplasm into two with each newly developing nucleus. Now complete the mitosis. Second slide of bacteria. Here we can see the lactobacillus bacteria, the bacterium kingdom Monera. What about Monera? Monera is the only kingdom with prokaryotic organisms. Prokaryote means premature, with premature nucleus. There is no nuclear membrane is seen. Kingdom Monera. Group Eubacteria, that means true bacteria. Eubacteria or true bacteria. What about lactobacillus? Here, in case of bacteria, the cell wall is made up of peptidoglycan. The cell wall is made up of peptidoglycan or in some cases muramic acid also. In some cases pili present. Now here we can't see the pili. In flagella may or may not be seen. Plasma membrane. The outermost layer is cell wall. Then inner to plasma membrane. It is lipoprotein in structure. It is made of plasma membrane is made up of lipids and protein. The genetic material that is scattered in the cytoplasm. There is no nuclear membrane, so they included in kingdom Monera. One circular chromosome made of DNA. One circular chromosome made of double helical molecule of DNA is located at the cytoplasm. That is known as nucleoid. What is nucleoid? A nucleus without nuclear membrane is known as nucleoid. Here the DNA or circular chromosome freely float in cytoplasm. In some cases of bacteria, apart from chromosome, in several species of bacteria possess more additional DNA called plasmid. In some cases, plasmids are seen in bacterial cell. Plasmid is an additional ring of DNA. What about cell organelles? Mesosomes helps for cellular respiration. There is no mitochondria present in bacteria. Mesosomes helps for cellular respiration. Ribosomes are seen. Only ribosomes are seen as cell organelle with 70 Sedberg unit. There are different types of nutrition in case of bacteria: autotrophs, saprotrophs, symbionts or parasites. Lactobacillus is saprotrophs. They feed on organic matter. That is the organic matter in case of the lactose of milk. Cellular respiration through mesosomes, asexual reproduction by budding, and sexual reproduction by conjugation, asexual reproduction by budding or binary fission. Here we can notice in case of lactobacillus binary fission is usual. Next, yeast. Yeast. Kingdom fungi. Kingdom fungi. Eukaryotes with a definite nucleus. And division. Eumycota. Division Eumycota. What about the cell structure? Cell structure. Here we can see the cell wall, mitochondria, like different types of cell organelles, cytoplasm, lipid granules as storage food material, nucleolus, nucleus, different types of cell organelles, nuclear membrane, definite nuclear membrane, and nucleus seen. So they are included in eukaryote. The yeast is a member of fungi. This is the one of the unicellular fungi. Yeast is the unicellular member of kingdom fungi. Cell is ovoid in shape. Distinct cell wall and nucleus seen. One or more vacuole in the cytoplasm. One or more vacuole seen in cytoplasm. Cytoplasm is granular. Granular cytoplasm is seen. Has glycogen and fat. Glycogen and fat or oil globules as reserve food material. Nutrition. In case of fungi, most of them are saprotrophic nutrition. They split glucose by the help of enzyme invertase or sucrase. With the help of enzyme, digestive enzyme invertase or sucrase, they split glucose molecule into ethyl alcohol, carbon dioxide, and energy. If from one molecule of glucose get 2 ethyl alcohol molecule, 2 carbon dioxide, and 2 ATP as energy, they utilize that ATP as energy, adenosine triphosphate. Reproduction, usually budding. Next, fungi, multicellular fungi. This is the structure of rhizopus or bread mold. Rhizopus or bread mold. Kingdom fungi, division Eumycota. What about the structure of rhizopus? We can see the stolon, rhizoid, sporangiophore with sporangium. This is the columella part. These are the spores. This is the ruptured sporangium with the columella and spores, matured spores. They usually develop on bread, chapati like food substances. They also split glucose and use the energy as ATP molecule for their development. Next, oscillatoria. Next, oscillatoria. Kingdom Monera. As like bacteria and different types of cyanobacteria are included in Kingdom Monera. Kingdom Monera means without a definite nucleus. There is no true nucleus seen in Kingdom Monera. Bacteria and cyanobacteria are the members of Kingdom Monera. Here we can see one of the cyanobacteria, that is oscillatoria. This is the colony of oscillatoria. We can identify one part, the apical cell, septum, septal disc, cell wall. What is the difference between bacteria and cyanobacteria? In case of bacteria, cells are usually small, but in case of cyanobacteria, comparatively larger cells are seen. This is the colony with numerous cells. Comparatively larger cells are seen in cyanobacteria. Bacteria may have flagella, but in case of cyanobacteria, there is no flagella are seen. Most of the bacteria are heterotrophs, but in case of cyanobacteria, they all carry out photosynthesis. All cyanobacteria members carry out photosynthesis in usual manner as in green plants and release oxygen. So they are oxygenic. In case of bacteria, sexual recombination by conjugation. In case of cyanobacteria, sexual combination has been observed in some cyanobacteria. Now we complete the first session of our botany practical, Viva and recorder session.